to Best Lives, the podcast that brings you the latest in pediatric research from BC Children's Hospital Research Institute. We highlight BCCHR investigators, clinicians, and trainees whose work is helping kids live their healthiest, happiest lives. Podcast guests share how their discoveries are changing the world of pediatrics and impacting families in British Columbia, the Yukon, and beyond. Whether you're a parent, healthcare professional, or just someone interested in the science behind kids' health, this podcast is for you. Tune in to stay informed and be inspired. My name is Kristen Hovitt, and I'm a research communications specialist for BCCHR. Joining me today are Dr. Sarah Merrill and Dr. Chaini Konwar. Dr. Merrill and Dr. Konwar co-first authored a research article titled Impact of Age-Related Changes in Buccal Epithelial Cells on Pediatric Epigenetic Biomarker Research, published in January 2025 in the journal Nature Communications.
I'm Sarah Merrill. I was a postdoctoral research fellow in Dr. Michael Kober's lab at BCCHR, and I am now an assistant professor and the PI of the Epigenetics and Psychosocial Interventions Lab at the University of Massachusetts Lowell.
Hi, I'm Chaini Konwar. I'm a bioinformatician in Dr. Michael Kober's lab at BC Children's Hospital Research Institute, where I have been working for over 5 years now alongside a fabulous team of data scientists who routinely works with high-dimensional epigenomic and genomic data. I have a PhD in medical genetics from UBC, where I was working on placental epigenetics and genetics with Dr. Wendy Robinson at BCCHRI.
For listeners who might not be familiar, why do cheek swabs matter in children's health research, and what can buccal epithelial cells, or BECs, tell us?
Great question. These cheek swabs are really useful for kids because they're not painful. They're not scary. They're not uncomfortable even, really. It's just a little plastic brush up against your cheek. We can use it in kids as young as infants. And we also have the benefit of cheek swabs not really running into the same kind of cultural and religious taboos that we find with both saliva and blood collections, which are other tissues that are commonly used for this type of research. So in terms of the actual collection of the sample, really useful for specifically a children's population and looking at children's health. And probably because of this, we have a large body of work looking at how we can predict things and how these samples can reflect things that have happened to children to improve their health. So things like exposure to stress or pollution or chemicals. We can also see that it can be useful for screening things like cancer or heart problems. Specifically, we study DNA methylation, which is a type of epigenetics, where we can look at these tags that are on the DNA. And they don't affect the genes themselves, but they can affect how the genes function. And they can also act as biomarkers of things that have happened in the past biologically. We can use these to look at children's health in terms of their actual experiences, as well as their likelihood of having a health problem in the future, or even if they are currently having a health problem at that moment.
Your study looked at cheek swabs from 12 pediatric cohorts, comprising more than 4,600 children, from babies to teenagers. Were there any surprises as the data started to come together for such a large and diverse group?
Yes, thank you for this question. Well, first, we would definitely like to thank our participants and our wonderful collaborators who graciously shared their cohorts with us. It really allowed us to sort of create this comprehensive database of cheek swabs from approximately almost 5,000 typically developing individuals and spanning across the entire pediatric range, which was from 2 months to 21-year-old. And some of the cohorts were even actually longitudinal, meaning we had measurements from multiple time points that allowed us to look at this data in a more nuanced way. Now, getting back to what was one of the most surprising or exciting bit that we found was initially at the start when me and Sarah started to like conceptualize or work in some of the preliminary analysis, what we sort of hypothesized, probably in a naive way, is that the linear decrease or like the decrease of buccal epithelial cell proportions with age, we just expected it to be in a linear fashion across all our participants, regardless of where they fall in the developmental window. However, one of the wonderful reviewers in Nature Communications pointed towards that there is sort of a subset of samples which did not seem to follow that trend. And while addressing the reviewer comments, when we carefully inspected our samples, we found that a subset of samples around the adolescents actually did not conform to the trend. And then we started working with some other sophisticated statistical approaches, which sort of allowed us to examine samples from different developmental periods a little differently. And that's when we sort of defined a transition from childhood to adolescence using a threshold of 10 years. Again, as stated in the paper, this transition is not about a single age per se, but more around a developmental period, which will encompass the ages at 10 as well. So we did our analysis for the 9-year-old cutoff and also an 11-year-old cutoff. And what we saw was that when we looked at this subset of samples from infancy to childhood, less than 10-year-olds, the decrease in epithelial cell proportions from the cheek swabs with age was more pronounced and stronger and was in a very like linear way. However, we did not see that in the adolescence, which is like around the pre-adolescence and the end of adolescence age. And also when we looked at variability, children in the adolescence showed much more variability in buccal epithelial proportions compared to the infancy to childhood. So all of these were pointing towards that, if you're looking at cell-specific associations with age, it is very important to consider the developmental period. And since then, we really acknowledged the relevance of the developmental periods and subsequently always perform sort of our analysis, appreciating that sort of distinction.
Thank you. And I think this question will probably overlap with what you just responded with, but one of your key findings is that the number of buccal epithelial cells drops as children age, then level off during the teen years. Why is this important, and what could it mean for interpreting epigenetic data in pediatric populations?
Right. Yeah. We do have some theories and speculation as to like why we're seeing the sort of differences depending on developmental periods. Some rodent data says that the morphology, the size and shape of the oral epithelial cells, that changes over time. There is a reduced proliferation. The size may increase, but there is reduced density. Another compelling sort of possibility is the presence of pediatric gingivitis, which is an inflammation of the gums reflecting bacterial challenge. And this condition is almost absent, or the presence of such bacterial species in the gingival tissue is almost absent in children, but are present in the mouth of the adolescents. So yeah, conditions like gingivitis or some other oral health events, oral hygiene, diet, use of orthodontic appliance apparatus, these all can contribute to the differences in the proportions that we have observed.
And we also see in the quite young part of our childhood, sort of 0 to 10 part of our samples, that have that significant and reliable decrease in buccal proportions, that this could also be related to normative craniofacial development, that as their faces and their heads get bigger, right? As they are wont to do, that this also affects the proportion of skin to immune cells, essentially, in the samples that we're seeing in a reliable way that you don't necessarily see in the adolescents, where you aren't seeing the same kind of facial development, but you are seeing a lot of variability from things like Chaini said, oral health, gingivitis, wearing braces, things like that.
So things like baby teeth, braces, as you mentioned, even how often kids visit the dentist might affect the cheek swab results. So what do you want other researchers to know about this, and even parents? What can they take away from this?
Yeah, I think that's a great question. For example, in our study, in one of the cohorts that had contributed data, we had information about both teeth brushing and dental visits. And what we found is we didn't see a significant difference in terms of, brushing their teeth more often. These are very young children. We are looking at, 2 and 3-year-olds. So this is not necessarily true for older children, but at least in young children, we didn't see a huge difference in the amount of teeth brushing. But generally in our population, they had good oral hygiene. But we did see a difference in people who did have regular dental visits in early life versus those who didn't. And the people who didn't have those regular dental visits were more likely to have more immune cells, essentially, in their mouth in comparison to the skin cells that we found. Very much in line with what some of the other research has found, for example, with braces, that because there are more opportunities for bacteria to hide in the mouth, more things going on in the mouth realistically, harder to get into those spaces with brushing and things like that, that you see a similar difference with more immune cells in the mouth and less of the buccal skin cells, right? And so I think what we would like our researchers who are also using these cheek swabs to know is that it's important to ask questions like this. Things like oral hygiene, dental visits, especially if you're working in populations where orthodontia is common. Not just braces, but, permanent retainers, nighttime retainers, right? Things that people wouldn't normally think to report to you as a potential health difference, that are things that you could then be able to control for in your models or account for, especially if you are concerned that it might be associated with variables that you think are important for your variables of interest. For example, a lot of epigenetic work is interested in the overall effects of socioeconomic status, which can encompass lots of things from income to education to your social position. And these can highly overlap with access to something like dental visits or orthodontics, which can be very expensive. And you wouldn't want to accidentally, essentially, confound these differences in cell types because of the types of immune cells and bacteria in the mouth with something that is actually an income-related issue.
You worked with something called an epigenetic clock, which estimates biological age as separate or different from a person's actual chronological age. How do these clocks work, and how does your study help make them more accurate for studying kids' health?
Great question. Epigenetic clocks, very popular these days. So an epigenetic clock, right, it measures your biological age or the age of your cells, essentially, right? And they use those patterns of tags, these chemical tags on your DNA that doesn't change your sequence, but can change the function of these cells, right? And cumulatively across many, many sites where these tags can happen, we can look at them and we know that they change reliably across people with age. And by measuring how much of that tag you have on your cells at any particular point in time and adding those together, we can estimate approximately how old you are biologically based on these epigenetic markers. And then we can compare that to your chronological age, your actual age based on your birthday. And that biological age can be higher or lower than your actual age. And that can reflect differences in your health, how your body is aging, how your body is developing outside of just your calendar number of years. And it is important to say that a lot of the work that has been done on epigenetic age is primarily in adults, right? Where they have found that having an older biological age in comparison to your actual age is a clear health risk. It puts you at a higher risk for many conditions, cardiovascular disease, diabetes, even an earlier death potentially. And a younger epigenetic age is always a good thing, basically, right? That being younger is good. But in kids don't just age, they develop, they mature. Getting older is an important part of the process. It isn't just a continuation into decline. It is less clear what being biologically older or younger exactly means in kids, but there is increasing evidence in children that being either substantially biologically older or younger, similar to both precocious and delayed development, is not necessarily healthy for kids. And that what you're really looking for is to be as old as you are, right? That your biological age is about as old as your actual age. And things like an experience of adversity, experiences of abuse, for example, high levels of stress have pretty consistently been associated with having an older epigenetic age in children, which, is also associated with health risks and things like that. So being able to measure this as a marker in children accurately can be very helpful, both in helping us understand how much of a difference is meaningful in what direction, which is a question we are still answering, and what does that really convey for risk. And in order to do that, we need the most accurate measures possible. So by accounting for these differences in the number of buccal cells to immune cells in your cheek swabs, as we recommend in this paper, we are able to have more accurate and more consistent measurements from these samples in kids, which will help us to be able to interpret and use and even develop new biomarkers better.
And Sarah has wonderfully explained the epigenetic clocks for us and their relevance. What I could just add a little bit is maybe think of epigenetic clocks as measuring the wear and tear on a pair of shoes. Imagine that as you walk in your shoes, they slowly start to show these signs of use, the creases and the wear on the soles. The more you walk, the more signs of aging one's shoes will show. So overall, what's important is the number of steps that have been taken wearing those shoes, not the calendar date of when you brought them. So similarly, as you age, the stress and adversities one's experience makes the cells older than our calendar age, which then increases our risk to various diseases. But this is, as Sarah kind of mentioned already, is relatively straightforward in the adult space. But as we have mentioned a few times now, the kids are not aging, they are developing. And so we're slowly starting to sort of understand what does it mean? What do these clocks mean, these epigenetic clocks in the pediatric space?
And you used one that the Konwar lab developed, right?
Yes.
Am I saying it right?
Correct.
Is it PedBE or PedBE?
Correct. Yes.
PedBE.
Okay.
Yeah.
PedBE.
Exactly.
That's the star of our paper, PedBE, which is pediatric buccal epigenetic clock, which is sort of the abbreviated version. And that is supposed to provide the most accurate estimation of calendar age or chronological age in pediatric cheek swab samples. And there are a multitude of clocks which are developed for adult aging populations, but clocks in the pediatric space is really limited, and PedBE definitely is one of those.
I'm going to ask another question that kind of talks about that transition around age 10. I know you talked about it already, but it's really an interesting part of the paper for sure. So your study highlights that developmental transition that happens around age 10. How might this transition in the context of buccal epithelial cell proportion influence how researchers design the clocks themselves and epigenetic studies?
I think I can start a little bit on this and then Sarah can chime in for sure. But what we are really saying using that cutoff is yes, we used a cutoff of 10 years to do most of our subsequent analysis. But really what we are emphasizing is that transition from childhood to adolescence. So it is not really about a single age per se, but it's that entire developmental period, which is why we tested most of our analysis using a cutoff at 9 or a cutoff at 11. But to sort of acknowledge that what is happening from infancy to childhood could be really different, as we observed, and distinct from what's happening in adolescence. We see this decrease in buccal epithelial cell proportions with age, very pronounced, very striking in a linear fashion from infancy to childhood. But then when we only narrow it down or restrict our samples to those around adolescence, which is pre-adolescence and end of adolescence, from 10 to 20 years, we don't really see that association. So just acknowledging that, when you're examining these cell type associations with age, acknowledging the developmental period and taking that into consideration is what we are suggesting.
We also saw that in the children who had a strong negative association with age and buccal proportion, with our youngest participants having the highest number of buccals in their cheek swabs, this was a strong relationship where your essentially cell type proportions in your sample, and therefore the methylation that you're able to measure, right? Because it is entirely cell type dependent, is completely confounded with age. So if you are looking for something that is like you would like to do a longitudinal study over the course of early childhood, very popular, a great question from a developmental origins of health and disease perspective. You definitely want to make sure that you are accounting for these cell type proportions, even when using cumulative biomarkers that were designed in these samples, because by virtue of looking over time, you have an inherent confounding with the cell type proportions that you'll have. And then alternatively, in adolescence, what we actually found was a lot of inter-individual variability. So there wasn't a strong relationship or any kind of confounding between cell type proportions and age. However, when we corrected for the differences in cell type proportions, we actually found stronger signals where those cell type proportions, because they were so variable in the adolescents, accounting for them essentially eliminated noise in our analyses that made our association stronger. And so actually accounting for these buccal proportions can be important even when you're not worried about it being a confound, because it may be actually responsible for some of the, the variability that you're not particularly interested in your variable of interest. And so modeling this, even though the relationship is quite different between children and adolescents, modeling with epigenetically estimated cell type proportions in mind, no matter where along the pediatric health spectrum you are studying, can be helpful for clarifying what the actual relationship between your variables are.
And I think I recall seeing an email from Dr. Stewart who was talking about OCD research. So is there a paper on epigenetics and OCD coming out soon?
Yes, stay tuned. So yeah, we had the opportunity to collaborate first with Dr. Evelyn Stewart from BCCHRI, where we looked at these OCD kids and looked at how, PedBE was able to sort of like capture a signal of that. And what we observed was a really cool observation, but also which we have already shown with children affected with autism as well. So what we saw was that cheek swabs, which are obtained from OCD diagnosed children, showed increased acceleration or were more biologically mature compared to non-OCD healthy controls. But what was most interesting was this magnitude of association was attenuated when we control for the proportion of buccal epithelial cells. Still significant, like the association was still there. It's just the strength or the magnitude was a little different. And the paper is that's coming up is more not looking at the clocks, but like another facet of epigenetic research, which is around epigenome-wide association studies. And I don't want to give the spoilers, but some cool stuff coming up soon.
What excites you most about where your research is headed next?
I am particularly interested, and this is probably not a surprise given the name of my brand new lab, in what this can mean for interventions, especially in kids. I have shown in some of my recent work, also with Dr. Konwar and Chaini and the team, in collaboration with some of our other great researchers on our team, such as Dr. Nikki Bush at UCSF and Dr. Justin Parent at URI, that looking at oral samples, both before and after interventions, and sometimes even much later, a year after these interventions have taken place, we can see differences when accounting, and especially when accounting for these buccal cell proportions the way that we recommend in this paper, that these interventions seem to be really genuinely making a biological difference for these children. For example, with Nikki Bush at UCSF, we looked at children with high levels of trauma who had experienced pretty significant adversity. Some had child-parent psychotherapy, which was an attachment-based therapeutic intervention that is designed to improve the relationships between caregivers and their children after traumatic events have taken place and increase the feelings of trust and safety again. And what we found was those who had the benefit of child-parent psychotherapy versus matched community controls who had experienced trauma but did not have the benefit of psychotherapy, they actually did not continue to have this biological age acceleration. where we saw that after the therapy, there was no increase in how much faster they were aging. However, the people who didn't receive the intervention were aging much faster than they were at baseline, and especially a year later, much faster. And that is also something that we have seen recently in a study where we looked at Jordanian adolescents with another collaborator, Dr. Rana Dajani, that we just recently presented at the Human Biology Association, where immediately after the intervention, we saw that those who didn't receive the intervention among these sort of high-risk Jordanian adolescents in Jordan, they were aging much faster. And those who received a sort of resilience-building, skill-building intervention were not. They were aging sort of the same as they were at baseline. So being able to use methods like this and know that the effects that we're seeing, especially longitudinally over time in pediatric populations, are not an artifact of, confounding with age or confounding with cheek swabs and these other facets and factors, makes us a lot more confident to be able to say, yes, we are seeing a biological effect here to the best of our understanding, and being able to implement these in biomarkers, at least for me, in intervention contexts, especially.
Yeah, and I'd just like to add that as a data scientist with a passion for pediatric epigenetic research, these are really exciting times to be in. And what I would really like to see is the application of PedBE and similar clocks in other developmentally relevant childhood disorders. Again, really coming back to the concept that children are developing and not aging. So in certain scenarios in kids, being biologically more mature may not necessarily be a negative thing. And yes, we saw that, OCD diagnosed children and in autism, they seem to be more biologically mature, but, we have just started at the tip of this iceberg and we need to know more. So I'm just excited how these tools will be applied across various pediatric conditions and health outcomes.
Very exciting. Thank you both. And a question that we're asking all of the podcast guests: how does your research help children live their best lives?
It's a great question. I think the honest answer is directly, it doesn't. But what we are doing and what work like this does, characterizations of these samples, how they act across populations, across developmental windows, and really fundamental aspects of the biology of how we use samples like this, how we look at changes over time, are important for building the foundation of what is relatively a young field in the scientific process, especially looking at it and how I usually look at it, which is really looking at social experiences and psychological experiences and how these can affect us at a biological level, especially in early life, to affect health, both in childhood and lifelong. And by building this foundation, I always joke, all the graduate students in the lab know that I say, we're really, putting in the building blocks that maybe no one will see many, many decades from now, but that will be the foundation on which things like individualized and personalized medicine, effective biomarkers that work across populations to predict health and predict treatments and outcomes will eventually be built on.
And now, we have this comprehensive database of 5,000 typically developing children, which will allow us to answer more of these questions and probably get at, how we can make a more sort of a translational difference to children and their health overall.
Thank you for listening to Best Lives. If you enjoyed this episode, please subscribe, rate, and share the podcast. Stay tuned for more inspiring stories from BC Children's Hospital Research Institute.